Okay, let's start the afternoon session. The first group of the afternoon session is Udayana group, the marine science group. The title of their talk is Natural Coast Shields, the interconnected role of the seagrass, coral reefs, and mangrove in achieving SDGs 14. Okay, if you are ready, please start your presentation.
Okay, good afternoon, everyone. Can you hear me?
Okay.
Okay, I hope all of us are in good condition. First of all, we would like to say thank you for the opportunity that given to us to present our presentation. The title of our presentation is Neutral Coastal Shields, the interconnected role of seagrass, coral reefs, and mangrove in achieving SDG 14. Next. Okay, let me to introduce the member of the Udayana University Marine Science Group. There is Frianata with her major Marine Science, and Niketut Cenikririn Pramaswari with her major Marine Science, and me, Gracel Marisa Sitorus, with my major Aquatic Resources Management, and the last is Loisa Cahyono with her major Marine Science. Next.
Okay, so first we're gonna talk about national overview of coastal reaches distribution. Indonesia is home to some of the world's richest coastal ecosystem with around 3.36 million hectares of mangrove, that's about 23% of global total, and over 51,000 kilometers squares of coral reef within the coral triangle, and 12 species of seagrass covering around 30,000 kilometers squares. The country plays a crucial role in regional and global marine biodiversity. These ecosystems support fisheries, protect coastline, and serve as habitats for endangered species like dugong and sea turtles. Their vast coverage make Indonesia a key hotspot for conservation and sustainable blue economy efforts. The picture on the left side of this slide is a real-life example of a healthy coral reef ecosystem. It was taken by a fellow student of marine science during a dive into Lamben, Karangasem, Bali, showcasing the beauty and importance of coral reef as part of Indonesia's marine heritage. And next, we're gonna talk about impact on ocean and marine biodiversity. Oh, sorry. Indonesia coastal ecosystem are critical biodiversity hotspot. Together, they form a complex and interdependent system that sustains marine life and coastal communities. Coral reefs support an estimated 1 million marine species, while seagrass meadow and mangrove forests provide essential habitats for fish, sea turtles, dugong, and crustaceans. These ecosystems are closely interconnected. Mangrove help trap sediments, protecting coral reef from siltation, while seagrasses serve as ecological corridor that links habitats and support species migration. However, they face growing threats such as coral bleaching due to rising sea temperatures, seagrass loss from eutrophication and dredging, coastal erosion and sedimentation from upstream deforestation also threaten mangrove regeneration zone. Losing even one part of this web can unravel the entire system. Protecting this ecosystem isn't optional, it's essential for the future of our ocean and millions who depend on them. Their conservation directly supports sustainable development goals 14 life below water. Next.
Okay, so there are three key coastal ecosystem. First one is mangrove, second one is seagrass beds, and the last one is coral reefs. This ecosystem work as natural barriers to protect coastal areas from environmental threats. So the first one is mangrove. With their dense root system, mangrove can help to reduce wave energy, prevent coastal erosion, and limit saltwater intrusion into the land. So the next one is seagrass beds. Seagrass plays a quiet but a powerful role. Seagrass can stabilize sediments and traps particles, which makes the water clearer and healthier for the marine life. And the last one is coral reefs. So coral reefs can act as a wave breakers, can absorbing up to 97% of wave energy before it hits the shore. So that is like having a natural seawall made of living organism. This ecosystem don't just look beautiful, but they are working hard to protect us every day. Next. So now let's explore how this ecosystem interact and the many services they provide based on the Millennium Ecosystem Assessment and IPBES or Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Service. There are four main types of service. The first one is regulating service. It's like a coastal protection, carbon storage, and erosion control. And the second one is supporting service. It's such a breeding and nursery grounds for many important marine species. And the third one is provisioning service. It's meaning they give us resources, especially food, like a fish and shellfish. And finally, the last one is cultural service because the ecosystem is have a beautiful view. This ecosystem provide aesthetic beauty, tourism opportunities, and help preserve local tradition. On the right side of the slide, you can see how mangrove, seagrass, and coral reefs work together, forming a connected habitat where each one supports the other. Next. Okay, so let's talk about the connection between ecosystem and climate change. So mangrove, seagrass beds, and coral reefs are not isolated. They form a multi-layer coastal bioshield as a natural defense system. So mangrove can filter nutrients, seagrass can trap sediment, and coral reefs depends on clean water to survive. So this connection make them stronger together than they are alone. Also, mangrove and seagrass is a part of what we call blue carbon ecosystem. They store large amount of carbon dioxide, which help reduce climate change. In fact, Indonesia hold up to 3.4 gigatons of blue carbon, which is about 17% of the world's total. Protecting this ecosystem not only keeps our coastal safe, but it also helps fight climate change by reducing storm risks, tidal floods, and erosion. Next.
Okay, now we're going to talk about coastal ecosystem trends. First, we have the coral reef. More than 85% of the coral reef in the Coral Triangle region, including Indonesia, are currently threatened by various stressors. Satellite data from Nias Island shows a decrease in reef coral coverage from 7,716 hectares in 1994 to 4,236 hectares by 2014, with a degradation rate of 174 hectares per year. And there are several restoration efforts being carried out. First, active restoration program using various coral transplantation technique. Secondly, target coral restoration in Nusa Penida, 253 square meter, and Gili Mantra, 550,440 square meter. And the last one is rehabilitation program under the Coral Triangle Initiative. Next, there's status and target for mangrove ecosystem in Indonesia. One, the Indonesian government aimed to rehabilitate 630,000 hectares of mangrove by 2024. Mangrove in Southeast Sulawesi show a high level of vulnerability to climate change. And the last one is program to plant 20,000 mangrove seedlings in Nusa Penida, 1.5 hectares of degraded area. And next, there's seagrass beds with their threatened status. Indonesia has seagrass beds that are globally significant but is facing widespread threat. The decline in the health of seagrass beds is due to changing environmental conditions. Next. There are some factors driving change, as in natural factors and anthropogenic factors. In natural factors, we have global climate changes. That is, the increase in sea surface temperature causes coral bleaching. Second, we have natural climate variability, such as El Nino, La Nina, and seasonal fluctuations. Third, we have geological processes, as in subsidence, natural erosion, and changes in the coastline. We're moving to the anthropogenic factors. as in coastal development, is land reclamation and habitat conservation, deforestation, increasing sedimentation and nutrient runoff waste. We have pollution, that is plastic waste and domestic or industrial waste. And last, we have destructive fishing activities, that is overfishing, destructive fishing practices 60% reduction in illegal and destructive fishing practices in 6 out of 7 conservation project districts. Next.
Okay, in this part, we're gonna talk about efforts and future potential, conservation and rehabilitation efforts. As part of an ongoing conservation and rehabilitation effort, Bali has launched several impactful initiatives to restore its coastal ecosystem. One notable example is the Bali Mangrove Forest Restoration Project in Tahura Ngurah Rai, which focus on rehabilitating degraded mangrove areas while enhancing their function as neutral carbon sinks. Additionally, the Coral Reefs Rehabilitation Program in Nusa Penida has gained international recognition for its success in coral restoration. Local NGOs such as Reef Check Indonesia actively collaborated with dive operators through coral gardening techniques to support reef recovery. Next. Okay. Emerging technologies and sustainable approach. As part of emerging technologies and sustainable approach, Bali has adopted innovative marine conservation strategies that combine ecological science with community engagement. One prominent example is the Nusa Penida Marine Protected Area, or we can call it MPA, which applies a zoning system and enforces ecotourism guidelines to balance biodiversity protection with sustainable economic use. In addition, seagrass conservation efforts in Sanur and Benua Bay demonstrate the use of community-based monitoring and transplantation methods, highlighting the integration of local knowledge with modern ecological restoration practice. Next. Okay, role of local communities, governments, and international organizations. In the context of the role of local communities, governments, and international organizations, Bali provides a strong example of multi-level collaboration in coastal resources management. Community-based groups such as the Lembongan Pengelolaan Sumber Daya Laut Desa Adat Pet in Nusa Penida play an active role in protecting marine ecosystem by working alongside NGOs and government agencies. The Bali provincial governments support the efforts through integrated coastal spatial planning and funding mechanism. At the international level, organizations like World Water Forum have conducted environmental education campaigns and capacity building programs in such regions such as Buleleng and Jembrana, helping bridge global conservation goals with the local action. Next. The next part is about future potential if ecosystem sustainable is maintained. If Bali maintains its seagrass, coral reef, and mangrove ecosystem, it can be a regional leader in the blue economy, especially through sustainable tourism, fisheries, and carbon trading. The ecosystem helps protect Bali's coastline from erosion and support food security for coastal communities. In climate terms, mangrove and seagrass areas in Bali are vital for blue carbon storage, contributing to SDG 14 targets, which is first is 14.2 sustainable management and restoration of marine ecosystem, and the second one is 14.4 for 14.5, which is protection of at least 10% of coastal and marine areas. Next. At the end of our presentation, thank you for your attention. To conclude, healthy oceans lead to thriving communities and together they ensure a sustainable future. Therefore, it is important to realize that our action today shape the world we will live in tomorrow. Just remember, what we save today save us tomorrow. With that in mind, let us keep working together to protect and preserve our oceans for ourselves and for future generations. If you all have any question, just ask and we would love to answer. Thank you.
Okay, thank you very much. So now the paper is open for discussion. Are there any question or comment? I have one simple question. So in your presentation, there's a term, blue carbon, appeared, but I don't know the meaning of the blue carbon. Could you please explain the meaning of the blue carbon?
Okay, let me answer it. Blue carbon refers to the carbon that is captured and stored by coastal and marine ecosystems, such as like in our presentation there is mangrove, seagrass, and salt marshes. This ecosystem absorb carbon dioxide from the atmosphere and store it in their biomass, which is roots, leaves, and stems, and it at and it in the soil. Unlike terrestrial forests, blue carbon ecosystem can store carbon for thousands of years, especially in underwater sediments where the composition is lower.
Okay, thank you very much. So are there any question, comment?
Please raise your hand because we still have 1 minute. Any question? Raise your hand. If not, maybe I will ask what the main threat or the main menace for this one. I mean, just now for like I mean like seagrass management in your area. May I know what the main threat of this one? Seagrass, I think this is for dugong, yeah. So any kind of problem or any kind.
Let me answer it. So the main threats on seagrass management in Bali, especially in Bali, is pollution, especially from hotels and resort, and land runoff, like sewage, and the most giving the most the most runoff is at wait, I forgot the so the main threat is the pollution, especially from hotel and resort.
Okay.
I think that's
So it's mean that a little bit difficult to invite dugong to Bali, yeah, because some conflict between just now from the hotel household and also from the landlord of the seaweed, yeah. Do you think so? Okay, thank you very much, Sensei. I think you can continue.
Okay, are there any question, comment? If no, let's move to the next presentation. Thank you very much. Okay, next presentation is given by Udayana Biology Group. The title of their talk is Potential of Bacteria from Mangrove Sediments as Biodegradation Agents of Low-Density Polyethylene Plastic Waste in Benua. So if we are ready, please start your presentation.
Okay, thank you for the opportunity. And hello everyone. Today we would like to present our study about potential of bacteria from mangrove sediment as biodegradation agent of low density polyethylene as known LDPE plastic waste in Benoa Bay, Bali. And this study supervised by Prof. DRDRA Retno Kawuri MPhil. And before we begin to the main presentation, let me introduce our research team. We are working on in this research together as a team from Udayana University. And maybe I will start with myself. My name is Muhammad Dimas Bayu Purnawirawan. I'm a biology student. And we also have Mas Lita. Mas Lita, please introduce yourself.
Hello everyone, my name is Maslita. I'm from Udayana University.
Okay, thank you, Mas Lita. And we also have Dwi. Please, Dwi, introduce yourself.
Hello everyone, my name is Dwi. I'm also from biology team Udayana University.
Okay, the next one is Putu Depa Prabanata. Please introduce yourself.
Hello everyone, my name is Putu Depa Prabananta. I'm pharmacy student from Udayana University.
Okay, the last one is Ayu Yeni. Please introduce yourself.
Hello everyone, my name is Made Ayuni Dwi Herisanti. You can call me Ayu. I'm a student from Aquatic Resources Management, Faculty of Marine and Fisheries, Udayana University. Thank you.
Okay, thank you. We move to the next slide. Okay, this research is part of PKM or Program Kreativitas Mahasiswa atau or Student Creativity Program. This program is organized by the Ministry of Education, Culture, Research, and Technology of Indonesia. And this project is encourage students to develop their creativity, innovation, and critical thinking by applying their academic knowledge to real-life problem. And there are several categories, and this research comes to PKM-RA, which stand for PKM Riset Eksakta or Exact Science Research Scheme. So the PKM-RA focuses on scientific research based on strong methodology and experimentation, and our research fits into this category as it involves isolating bacteria, conducting biodegradation tests, and applying molecular technique to address environmental issues. And the background of our research comes up from major problem in Indonesia, which is plastic waste. It's around 64 million tons produced annually, and 3 million tons end up in the ocean. Among plastic type, low density polyethylene or LDPE is the most commonly found in marine environment. And LDPE is resistant to degradation, leading to accumulation in mangrove ecosystem and damage to biodiversity and substrate quality. And mangrove areas such as Benoa Bay, Bali are prone to plastic accumulation due to their dense vegetation. And research shows 56 to 97% of plastic debris gets trapped in mangrove roots. And sediment can accumulate plastic waste up to 50 centimeter depth, severely harming mangrove health. And that's all the background. And we have conducted field observation about plastic waste finding in the mangrove ecosystem of Benoa Bay. So as we can see in this picture, we know that there are plastic debris in the Benoa Bay area, including LDPE film, plastic bottle caps, rubbers, and food container on wick and sand in mangrove area. And there is a close-up of a discarded plastic food package being held in front of mangrove habitat. And the next side is plastic waste such LDPE film, sachet, and packaging material partially buried in the mangrove sediment. That's all documentation our observation field. there are our alternative solution based on research gap. So the physical and chemical degradation method are energy intensive and may produce harmful byproduct. So that's so biodegradation using bacteria is a safer and more sustainable alternative. It's supported by previous studies have shown Pseudomonas and Bacillus species can degrade plastic by 2.7 until 8% over 3 months. There are limited research on the potential of mangrove sediment bacteria to degrade plastic, especially in Indonesia. So the study focuses on exploring new bacterial species from mangrove sediment with potential for LDPE degradation. And our objective research to explore the biodegradation potential of bacteria from mangrove sediment in Benua Bay, Bali, and identify bacterial species capable of degrading LDPE plastic. So that's our alternative solution. And we move to low density polyethylene that will explain it to Mas Lita. Please, Mas Lita.
Next, the literature review in research, low density polyethylene or LDPE. LDPE is a thermoplastic polyethylene discovered in 1933 and widely used commercially. LDPE offers a combination of low cost, ease of fabrication, and balanced physical properties, making it suitable for various applications such as film, coating, molding, and wear insulation. LDPE is widely used as a polymer film material for everyday packaging, such as plastic bags and a pouch. Next. Plastic pollution in mangrove sediments. Mangrove area are highly vulnerable to plastic accumulation, especially in the sediment. The buildup of plastic waste interferes with root respiration, potentially leading to the death of mangrove trees. Description of associated marine life and an increased risk of coastal erosion. Common types of plastic pollution in mangrove include plastic straw, ropes, fishing lines, rubber band, bottle cup, rubber, and diverse, which often arrive from river current and human activity. Next, plastic biodegradation. Bacteria such as Idonella saccharomyces, Acinetobacter baumannii, and Pseudomonas species product enzymes like esterase, glutinase, and lipase that break down plastic polymers. These enzymes convert plastic into microbial biomass and gases, making the process eco-friendly. And the next,
All right, for the methodologies here, for the overview of methodology that we use, the first one is sampling of mangrove sediments in Benua Bays, which is the sample. After we collected the samples, we do the isolations or the bacterial isolations and the selections. And after that, we are moving to inoculum preparations to get more bacteria to be cultured further. And then we'll have the LDPE venom preparations to see the biodegradation effects from the bacterial activities, and then we'll move to the observations of biodegradation test for 44 days. And then after that, we were looking for the weight loss measurement with gravimetry test. And then we are going to observe the biodegradation effect by the bacteria's activities by the scanning electron microscopy or SEM to see any ropes, any rip of the plastic because of these bacteria. And then the last one, we are looking for the molecular identifications with 16 as rRNA. So for now, a little bit disclaimer, our process of research is still in the second phase here. After all of the 8 phases that we have, 8 stages, we are still in the bacterial isolations and selections, and we don't have our final results yet, but we are going to explain all of the results for now, the recent research, to all of you. So that's the overview of methodology. We are moving to the current progress. So this is our current progress. This is the sampling of mangrove sediments, which is sediment samples collected using the proposed random sampling. We are looking for the areas that there are the accumulations of the plastic, more accumulations of plastics. So we choose that area to propose a sample there and propose a random sampling from the 5 stations or 5 points in the mangrove area of Benoa Bay Bali, suspected to be contaminated with LDPE plastic. And sampling that we use is conducted as the 5 stations, which is the first one is Mangrove Information Centers or MIC, and we are moved to Serangan Mangrove, Benoa Mangrove, Kampung Kepiting, and Kelan Mangrove. It's station sample in triplicate, or we are collected 3 points of samplings in one locations, and about 100 grams of sediment collected at a 10 to the 15 centimeters depth, stored in Ziploc bags, and then placed in a cool box at 4 degrees Celsius and brought to the lab for further analysis. And we're moving to, this is a little bit of the documentations of our process, and this is me here. And there is DEPA also in the left side of the pictures, DEPA from pharmacies. And this is the process of bacterial installations and selections. So the 10 grams of sediments that we have collected before, the samples is mixed with 90 milliliters of 0.9% saline solutions, then incubated at on a shaker at 37 degrees Celsius, which is the ideal temperatures for it, for 30 minutes. And serial dilutions are made up to 10 to the negative 10 to the negative 6. Its dilutions is plated in 0.1 milliliters onto petri dishes containing mineral salt medium, which is the select media for this bacteria, that's supplemented with 1 gram polyethylene glycols or PEG, and will be incubated for 48 hours at temperatures at 40 degrees Celsius. So the results and discussions will be explained by Dimas once again. So for Dimas, time is yours.
Thanks for the opportunity, and I will explain about the result and discussion. So this table shows the total plate count of bacterial isolate from mangrove sediment at 2 different locations, which is the Mangrove Information Center and Kampung Kepiting. So as we can see, several samples, especially from the Kampung Kepiting site, so very high bacterial growth. For example, sample KKT1 had the highest number of colonies with 273 at 10 to the negative 2 dilution, and 32 at 10 to the negative 3. So meanwhile, some of the MIC samples, such as MIC T2, showed moderate growth, and many samples at 10 to the negative 1 dilution were labeled as TBR or 2 numeric 2 count, so which mean indicate extremely high microbial presence. And interestingly, in some of the petri dishes, we observed clear zones forming around the bacterial colonies, as we can see in this picture. And this might indicate potential LDPE degradation activity by this isolate, something we aim to explore further in the next step of our research. That's all from me. And continue to Maslita. Maslita, please.
Okay, thank you, Dimas. Next. Okay, expect outcomes in our research. First, discovery of bacterial isolates from mangrove sediments that are capable of degrading LDPE plastic. Second, contribution of scientists' knowledge on microbial-based solution for plastic pollution, especially in mangrove ecosystem. Third, foundation for future development of eco-friendly biodegradation technology using local microbial research. And next will be explained by Dwi.
All right, thank you, Maslita, for the explanations for our expected outcomes. Before it, there is some things that we skipped before, which is going to be explained by me. So what's next? What is this next steps that we would like, we're looking for? So the next step is, because now the process is still in the isolations of the bacteria, which is the potential bacteria, we are going to move to inoculum preparations. So the selected isolates are cultures in modified TSB with 30 gram per liter NaCl or natrium chloride at 30 degrees Celsius for 24 hours on a shaker. The cultures are centrifuged at 6,000 RPM, 15 degrees Celsius for the temperatures and for 10 minutes for the periodic. To separate the cells, wash twice with sterile 0.9% of saline and adjust it to a final concentrations of 10 to the 6 until 10 to the 8 CFU per milliliters. So for the consortia, isolates are mixed in equal volumes and antimicrobial compatibility is tested using well-diffusion assays to ensure no mutual inhibitions. And after the inoculant preparations, we are looking for LDPE preparations, just like what we have explained before to you in the method and overview of the method before. So LDPE preparations, which is the LDPE plastic bags, are cut into 1x1 centimeter pieces, sterilized in 70% alcohol for 30 minutes, and rinsed with sterile water and air dry under UV light in a laminar flow cabinet for 30 minutes. And the dry weight is measured using an oven at 80 degrees Celsius for 24 hours, and each piece is level. So before we start to forget, we have to label it. And the next step after two of that is we are looking for the gravimetric weight loss. So here is the explanations of gravimetric weight loss measurements after the biodegradations. So LDPE films are dried under vacuums at room temperatures until this constant weight is achieved. Gravimetric loss is calculated with this in percentage is equal to initial weight minus the final weight per final weight times the 100. So after the gravimetric weight loss measurements, microscopic or SEM. So after biodegradations, the LDPE surfaces must be 9.8 nanometers and observe under SEM or scanning electron microscope at 5,000 times 2 100,000 times multiplications to identify holes, any cracks, and or degradations marked caused by these bacterial activities. And then the last one here is the molecular identifications in our method. The last one is the identifications for the molecular. So we want to know which bacteria cause this biodegradations of the LDPE plastic in the mangrove area. So the molecular identification is conducted to determine the bacterial species involved in LDPE degradation using the 16S RNA gene as a marker. The process begins with DNA extinctions from selected bacterial isolates and followed by PCR amplifications of the 16S rRNA gene. The resulting amplicons are verified through gel electrophoresis, then sent for sequencing. The obtained sequences are analyzed using BLAST or Basic Local Alignment Research Tool against the NCBI database to identify the closest known bacterial species based on genetic similarity. So that's the next steps and explanations of our method before that we have been explained, and the expected outcomes have been explained too by Mastita. And here are the conclusions for our presentations today from Udayana University Biology team, which is the bacteria isolated from mangrove sediment, show high colony counts, especially from the KK side or double K, which is double K is referred to kampung kepiting. So clear zones around some colonies suggest potential LDPE degradation ability. This highlights the mangrove ecosystems as promising source for the plastic degrading microbes. So this is our team and our supervisors in the middle of us, Prof. Retno, and this is the face of us as a biology team in Udayana University. So thank you very much. That's all for our explanations, and we're looking upward for the discussions. Thank you.
Okay, thank you very much for your nice presentation. And now the paper is open for discussion. Are there any questions or comments? Okay, so I have a question. So in your presentation, you are focusing on bacteria as an agent of the plastic degradation. But I think the other agent exists. So the fungi is other agent of the degradation of the biodegradation of the organic materials. So my question is, why you focus on the bacteria instead of the fungi? I think the marine fungi exist in the mangrove. So what is the merit to focus on the bacteria instead of the fungi? That is my question.
Thank you for the questions. We seek times for a minute to discuss it,
Yeah.
and we
Okay.
will
Okay.
answer the
Okay.
questions. Thank you. All right, so why we choose the bacteria more than the fungi as our research? So the first one is the background of the research. In mangrove ecosystems, we think that bacteria grow faster, which is they have the various species in the mangrove. And besides that, the bacteria's metabolism is better studied and also mapped. And besides that, bacteria are more genetically trackables, and we can say that after the observations, the bacteria grow faster, so we can identify it as soon as possible, which is it is related to the deadlines of the research.
Okay, thank you very much. So, are there any question, comment? If no, let's move to the next presentation. And I'm sorry, I'll leave this workshop due to my health condition. So the chair is switched to Professor Bibin from here. So the Professor Bibin, please.
Okay, thank you very much, Professor Toh Odajini. Okay, okay. Okay, everybody. So now I am going to continue. So I think I will give time to the next group. Yeah, I think KGU group 2. Yeah. Title is Sustainable Wetland Management, the role of tree cutting and wood reuse. So time is yours. So I will let group 2, please make some preparation.
Hello everyone. Today our group will talk about how cutting trees and reusing the wood can help protect wetlands, especially in Satoyama areas. We joined an activity to manage a local wetland and learned a lot about why cutting tree is necessary and how to reuse the wood in sustainable ways. We want to share what we learned with you today. First, what are wetlands? Wetlands are areas with lots of water. Many rare plants and animals live there, so they are very important for biodiversity. But if wetlands are not well managed, their ecosystem can break down. That's why we need to take care of them. Now, let's look at what's happening in Satoyama. Satoyama is a semi-natural area where people and nature have lived together for a long time. But recently, many Satoyama areas are being left unmanaged. As a result, evergreen trees grow too much. That makes the forest too dark, and the plants and animals that need sunlight start to disappear. In the end, the forest loses biodiversity. This is a big problem, especially for wetlands inside the Satoyama. So, what can we do? We learned that cutting some trees is actually necessary to protect wetlands. It may sound strange, but it's true. By cutting evergreen trees, we bring back sunlight. This helps living plants and animals come back. In short, cutting tree is not destroying nature. It's protecting it. Here is our presentation outline. First, why tree cutting is sometimes necessary. We will explain the difference between evergreen and summer-green trees, and why we choose to cut certain trees in the Saraki wetland. Second, how the cut wood was reused. Next, we will show how we reuse the wood in practical ways, such as wood chips for trays, benches, and so on. Third, other creative and sustainable explains. We will also introduce additional ways to use felled trees, like turning them into biomass foods or purposing them into tools, art, and learning materials. Fourth, environmental education. Finally, we will explain how the cut wood became a tool for education. Through workshops, upcycling projects, art exhibitions, and wood education for all ages. Now, let's start with part 1.
Why trees cutting is necessary. I will talk about the difference between summer green trees and evergreen trees, the type of tree we cut down, and the reason why we did so. Please note that we didn't cut down all the trees, only selected ones. In this picture with the white frame, you can see evergreen trees. The feature of evergreen trees is that they have leaves all year round. Their leaves are thick and hard, and their surface is often shiny. Example, evergreen trees include Japanese red pine, Japanese cedar, and the camphor tree. Because of their dense foliage, they tend to block sunlight even in winter. This picture shows a summer green tree. The main feature of summer green trees is that their leaves fall in autumn and winter. Their leaves are soft and thin, and the overall appearance is lighter. Examples are oaks and sawtooth oaks. In winter, because they lose their leaves, more sunlight can reach the ground. The tree we cut this time was an evergreen tree. We identified it by checking the features of its leaves, especially the hardness, thickness, and shiny surface. When we weren't sure, we asked the Moribito volunteers for help. We also noticed that evergreen leaves feel warmer when touched due to their density. Now I will explain the reason for cutting the evergreen trees. Main reason this decision was made by the Moribito volunteers based on the needs of the wetland. There are too many evergreen trees in this area, especially in the Saraiki wetland. Because they grow densely and keep their leaves all year, they block sunlight from reaching the ground. By cutting them, we can improve sunlight for wetland-specific plants and help new ones grow. Also, since summer green trees are here, this helps balance the variety of trees in the area. In some other wetlands in Kyushu, summer green trees are sometimes removed, but here we focus only on evergreen trees. Next, I will talk about utilization of felled trees in the Saraike wetlands. We mainly utilize them in three ways. First, we chip the evergreen trees and use the wood chips to improve the walking trails. In the mountains, trails can become muddy and slippery after rain, which makes walking difficult and even dangerous. By laying down wood chips, we were able to absorb moisture and provide better traction. This made the trails safer and more comfortable to walk on, especially on rainy days or just after it had rained. Second, we used the harder tree to make benches. Before cutting down a tree, we first decided the direction in which we wanted it to fall. Then we tied a rope to the tree and made a knot called a receiving cut in that direction. This involved cutting straight into the trunk, followed by a 45-degree angled cut to form a triangle knot. From the opposite side, we made a straight cut known as the back cut. When we pulled the rope, the tree fell exactly where we intended. After felling the tree, we cut off the branches and smoothed the surface so it would be comfortable to sit on. Finally, we fixed the bench in place using clamps, and our handmade bench was complete. Third, we cultivated shiitake mushrooms. Shiitake is a popular and flavorful mushroom used in many Japanese dishes. We harvested shiitake that had grown on honara logs that had been inoculated with mushroom spores by Moribito volunteers in advance. Besides these three main uses, we also roasted some of the leaves and steeped them in boiling water to make tea. The tea had very good aroma.
I want to talk about past environmental education and message for the future. Peat trees with irregular shapes can be used as biomass fuel, like wood pellets, while burning wood pellets release CO2. It's considered carbon neutral since trees absorb CO2 as they grow. Japan promotes their use toward its uh, 2050 carbon neutrality goal. Traditionally, winter lodging helped forest health and provide firewood or charcoal. Uh, Recently, poor forest management has led to oak with spread beetles. Reviving traditional tree felling and food use may help address this issue. Second, I want to talk about actual use cases. From here, I would like to explain some specific ways to use wood. Broadly it is divided into graphics and stationery. For example, pencil, eraser, pottery, fabric. Third, I want to talk about consulate activities. First, tree life workshop. Students examine tree rings to learn the tree's age, origin, and reason for cutting. They make wood name tags or key dates to update the tree's value. Second, upcycle project with cut wood. Students use cut wood to build useful items like planters, benches, or birdhouses for local parks or schools, learning resourcefulness and sustainability. Third, forest art examination. Students create art from branches, leaves, and wood, showcasing their work and connecting with nature's beauty and texture. While nature, nature's beauty, while fostering creativity and environmental care. Uh, and in addition to wood learning class. Finally, uh, I want to talk about main usage examples. Particle boards and fiber boards are made by binding wood scraps or fibers with adhesive into uniform boards. Those sensitive to moisture due to glue. They resist wrapping and cracking and can be made in any size or thickness. They are made use and as indoor furniture bases and roofing materials. I finish. Next.
Art trees are not just wood. They can teach us about nature and how to take care of the environment. I will explain about that with four ideas. First is Tree Life Workshop. In this activity, students look at the tree rings of a cut tree to see how many years it lived. They also learn where the tree grew and why it was cut down, using maps and pictures. After that, they can make a name tag or keychain from the wood. This helps them understand the life and value of a tree. Second is Upcycle project with cut wood. This project uses cut wood to make useful things for the local area. For example, students can build planters, benches, or birdhouses and place them in parks or schools. They can see how a tree can change into something helpful and learn about using natural resources in a good way. Third is forest art exhibition. Students use tree branches, leaves, and wood pieces to make art. They can create anything they like and show their work in a small art show. This activity helps them feel the texture of wood and think about the beauty of nature. It also builds creativity and care for the environment. Fourth is wood learning class. This program changes by age. For younger children, they play with wooden toys and feel the warmth of wood. For older students, there are lessons about why we cut some trees and how forests are important. After learning, they can try a simple wood project. It is a fun way to learn with all 5 seasons. In conclusion, using the trees like for learning is a great way to grow our hearts and minds. We learned that cutting trees is not always bad. Sometimes it is necessary to protect the wetlands and help more kinds of plants and animals grow. We carefully selected which tree to cut, focusing on evergreen trees that block sunlight. After cutting, we reused the wood in many creative ways, like making benches, chips for trails, and even growing mushrooms. We also use the trees for running. So activities like the tree life workshops, art project, and wood upcycling. We could understand the value of trees more deeply. These programs helped us think about nature, sustainability, and how to take care of the environment. Thank you for listening.
Wait a moment. Oh, okay. So thank you very much. So we still have 3 minutes. So any question? So we open to the floor. Any question, please? The other group or from the other university, if you would like to ask to the KGU group too. And you can directly maybe, or maybe you raise your hand. Okay, from UiTM, Shamila Aina Kamarudin, please, directly you can ask, and directly also KGU will answer. Please come here.
Can you hear me?
Okay, please.
Okay. Just now you mentioned about boiling leaf tree to make a tea. So I just curious to know about what the nutrition that you can get from the tea. What the purpose of doing that tea?
What the purpose of it? So you ask to the group 2 that this tea, okay?
Yeah.
Yeah, okay, please you answer.
Thank you for your question. We use the roasted leaves of cut trees to make tea. This was not mainly for food, but for experiencing how we can enjoy nature in different ways.
So do you mean that they don't give any benefit to us, just about to enjoy the nature?
Yeah. So get the energy.
For you mean like forgive the energy for our body? Just it.
Yes.
Okay. Thank you.
Thank you.
Thank you. Okay, we still have I think time is over. It's already 2:46. So I think thank you very much for your question. And I think I'm going to give to the other group. Yeah. Thank you very much. Thank you, group 2 from the KGU. Okay, I think from UMMUL, I think please open.
Good afternoon, everyone.
Okay, it's nice.
We are from UNMUL Group, from Faculty of Forestry and Tropical Environment, Mula Warman University. So the title of our presentation today is The Potential of East Kalimantan Plants as a Natural Guide to Protecting Nature. So this is our member. One of them is master student. As we know that East Kalimantan has a tropical rainforest which have mega biodiversity, and the diversity of plants in the tropical rainforest of East Kalimantan is a source of life for local communities, and local communities usually utilize forest products to turn plant parts such as leaves, stems, bark, root, or even the waste of into natural dyes. And the advantage of natural dyes, including this element, first is eco-friendly. So because the natural dyes don't have negative impact for the environment, unlike the synthetic dyes, which can pollute water and soil. Next is enhancing local wisdom. Because East Kalimantan has a lot of cultural heritage, such as woven sarung or sarung tenun, eco print, and doyo weaving or ulap doyo, where the fabric fibers are dyed using natural dyes. Third is eco-entrepreneurship. Because using natural dyes not only giving color to food and fabric, but also contain phytochemical compounds, and that can be again of antioxidant, antibacterial, antifungi, etc. Next is in accordance with the focus on the SDGs alignment, this theme is associated with 4 key elements that align with the objectives of the Sustainable Development Goals. Number 6, clean water and sanitation. Because synthetic dyes which are currently used have a negative impact for the environment, and it can polluting the waters, especially in rivers. So, by using dyes from biomaterials, water pollution will be reduced. SDGs number 9, industry, innovation, and infrastructure. So the natural dyes are an innovation to support sustainable industry by paying attention to their impact on the environment. SDGs number 11, sustainable cities and communities. Because it helps preserve traditional knowledge and local culture, and encourage sustainable urban development by reducing dependence on hazardous chemical and industrial waste. And SDGs number 12. Natural dyes can reduce dependence on synthetic chemicals that pollute the environment.
The next slide is we're gonna introduce and explore several plants from East Kalimantan that have been proven as effective natural dyes, each with unique phytochemical compounds and health benefits. First, we have ketapang, or often called the tropical almond offers a tannin. The tropical almond offers a tannin-rich extract and producing stable dyes in acidic conditions. It also contains compounds with antimicrobial and antioxidant effects. Another plant is kasumba. The part of this plant that is used as natural dyes is the seeds. Kasumba seeds are rich in bixin, which is a carotenoid pigment that serves as an effective natural dye, used as a vivid orange and red dye, particularly in batik, and also serves a medicinal plant with detoxifying properties and strong antibacterial effects. Next is Swietenia macrophylla, or mahogany, has been widely planted across the island. Its bark and wood are rich in flavonoids and tannins, which make them suitable as a source of brown and yellow dyes. Beyond aesthetics, these compounds contribute antioxidant and antibacterial benefits. Dye extract from mahogany have been tested on cotton and silk fabrics, showing not only fastness but also antimicrobial protection.
Okay. For the next, Eusideroxylon swageri, commonly known as ulin, is an endemic species to Kalimantan and one of the most iconic hardwood trees in the region. Recent studies have shown that the sawdust and bark of ulin can be extracted to produce a reddish
to brown dye that has high color fastness, especially when properly fixed on fabric. These extracts are rich in tannin and flavonoids, which also give the dye antibacterial properties. Methanol extract from its bark in particular have shown strong inhibitory effect on bacteria Staphylococcus aureus. And the next, another local plant is Sorea macrophylla, commonly known as tengkawang. This tree is also endemic to Kalimantan, and its seeds have traditionally been used in weaving craft to produce a yellow base color. Boiling the seed releases dye compounds such as flavonoid and tannin, which also carry antimicrobial and antioxidant effect, in addition to its use in textile dyeing. Tengkawang oil is known for its moisturizing properties and is already used in cosmetic product. And next, we present Durio-cutejensis or lai. This fruit species is a close relative of the common durian and is native to East Kalimantan. While the fruit is well known as local delicacy, the leaves of the plant have also been utilized as a source of natural brown dye for batik production. The ethanol extract of lai leaves contains a mix of tannins, alkaloids, triterpenoids, and flavonoids. This compound give the extract both antioxidant and antibacterial properties, making it another excellent candidate for sustainable dyeing solution.
Next. Okay, next we have Chang or sappanwood, known for producing red dye through the compound brazilin. It has strong antioxidant and antibacterial properties. Next. Okay, and the last one, another plant with significant potential is Melastoma malabatricum, locally called karamunting or senduduk. It is a white shrub that grows abundantly in the forest and is often overlooked. Its fruits are edible and rich in anthocyanins, the pigments responsible for their deep blue-black colorations. In addition to their use as a natural dye, the fruits have been shown to possess strong antioxidant capacity. This plant also exhibits antibacterial activity across all of its parts, making it a multifunctional species with both nutritional and industrial applications. And next. Okay, next again. In conclusion, the forests of East Kalimantan are not only crucial for ecological balance, but also hold vast potential for sustainable innovations. The native plants we have presented today demonstrate how local biodiversity can be preserved while supporting eco-friendly industries, cultural heritage, and even health sectors. By promoting the use of these natural dye sources, we can reduce reliance on synthetic chemicals, minimize environmental damage, and create new opportunities for local communities, especially those involved in traditional crafts and small-scale enterprise. Thank you very much for your attention. We hope this presentation has broadened your perspective on how local flora can become agents of change in the movement toward sustainability. We welcome any question or thoughts you'd like to share.
Okay, thank you very much. Yeah, so now I think we still have a lot of time for discussion. There will be around 12 minutes. Yeah. Okay, so I will open to the floor. So maybe the other student from the other university if you would like to ask, this is your chance to know about the potential, natural potential, natural protection, or natural, what is it, the some kind of the potential or benefit from the natural resource, yeah, from the Kalimantan. So I think I would like to open this one. Okay, anyone would like to ask? This is your chance. Maybe I will start from my side. Okay. And please prepare if you would like to ask, or maybe you will continue. Maybe if you don't understand from our discussion between me and the participant from the UMMUL, Universitas Mula Warman. Concerning the natural dyes, in this case, I would like to ask about what is it, durable of this coloring, yeah. If we compare with the synthetic, yeah, or from the chemical, so which one that it is the durable, which one the longest one, yeah. This is one I would like to ask. And any additive that maybe I mean the user would like to add, yeah. And how about the price if it is released to the market, which one the very I mean, little bit. I think the customer habit. They would like to have the cheapest one. Can what is it? Natural dye compete with this situation? Okay, I think I released three questions to you. Please, I hope you can understand, yeah, my questions. If not, I will repeat again.
Wait a minute, sir.
I think waste from the ironwood is one of the cheapest because we use waste.
Okay, then.
Okay, I will continue the answer from my friend, especially Safirna. I think the natural dyes can last a long time if added with natural preservatives such as like aluminium sulfate can make natural dyes have less along color in the object what I want to use. And then about the price, I think about the price of natural dye can fit the market. And I think positively if we use the natural dyes can minimize environmental damage and create new opportunities for local communities, especially those involved in traditional crafts and small-scale enterprise. So that's the answer, sir, from us.
Okay, so I think I can conclude that there is the cheapest one, yeah, from the what is it, natural dyes, yeah, but I forgot the name, yeah, but okay, no problem. So there's available, and then the durable also, not so very different, yeah, with the chemical. Okay, I can get it, and there's no any addition, yeah, chemical. Oh, just now you you mentioned about what is it? So sulfate acid. So what does it mean? It's mean you ate this one for as the additive for to the natural dyeing, or just natural dyeing indirectly? Please, can you repeat?
Okay, about the aluminum sulfate, it's for additive on the natural dyes to make it strong color. As we know, aluminum sulfate, it's from chemical organic too. So it's safe for the environment.
Okay.
And I think the price of the aluminum sulfate is the cheapest one. Yeah.
Okay, thank you very much. I can get and I can understand. So it's mean the organic chemical, you add this one organic chemical just to enhance the color. Okay, so thank you very much. So we still have 4 minutes. So please, I will give to the floor. So who know or to audience who from the other university, maybe you would like to ask, or maybe from Malaysia to compare with your experiences for KGU student or from the other university, maybe you would like to get more information directly. No, from Mula Warman, inform or maybe give some promotion. Okay, from UiTM will ask Shamila. Samila, please, you can directly ask.
Hello. I just curious, how do you extract these dyes for the commercial use? That's it.
Okay, thank you. How to extract?
Okay, thanks for the question. Maybe can give can wait sorry, wait a minute for discussion.
So we will extract the plants with aqueous solvent because it's the simple and cheaper solvent, I think.
So you doesn't extract in laboratory or factory punya like factory punya what they call method. So you just doing such a thing to extract, not in laboratory or huge sector like that.
Can you please repeat the question?
I just asking that you is it you extract the dyes in laboratories or in the factories for the commercial use? And you can explain a bit how you extract if you know. If you don't know, it's okay.
We doing the extraction process in laboratory, so the small small pieces.
Yeah, I think Shamila would like to make some confirmation from you that it is you follow laboratory method or you just follow from the local people method or traditional method, only like this maybe, yeah? Is it correct, Shamila?
Yeah. Absolutely.
How to extract this one? You follow the laboratory.
Okay.
Okay.
Excuse me, I will continue the answer from my friend.
Okay, please.
So the method is the simple in our laboratory, which is to boil the sample as the community does. And then from the boil the sample, we can research about the phytochemical and bioactivity character. So the method is simple, as the community does.
What do you think, Samila? Is enough or maybe you still curious?
I think it's okay.
Okay, thank you very much. So because also time is over, so I think it's already 3:11. Thank you very much for your presentation from University of Bula Warman. So next, I'm going to give next group. I think this is the last group, yeah, from KGU, group 3. So I think The time is yours, please.
Good afternoon. We are Group 3 from Kansai Gakuin University. Today we explore a fundamental question. What is essential for sustaining Satoyama conservation today? Satoyama used to be a place where people and nature coexisted. It provided fuel, food, and resources essential to daily life. But today, due to the energy revolution, depopulation, and urbanization, human connection with Satoyama has faded. Many areas are now abandoned. Okay, so we ask, in today's society, what is truly essential for Satoyama to be conserved permanently? This is not just a question of nature. It's a question of people value and relationship. To answer this, we combined academic research with real field experience. In today's presentation, we will explore the current state of Satoyama, share our field experience, introduce the idea of a win-win relationship, present real-world examples, both in Japan and abroad. Finally, reflect on what Satoyama means today.
Introduce you the reality of Satoyama and social background. For a long time, Satoyama has been an intermediate space where people and nature support each other, and it has been closely related to our life. Satoyama was used for fuel, fertilizer, food, and fuels. In addition, it was a place where people and nature coexisted. Satoyama is closer to the fields than nature. It is an environment established after the destruction of nature, secondary nature, an artificial environment, but it is an important environment with high biodiversity. However, if Satoyama left unattended, it will become rough and the ecosystem will change and the species will decrease. On the other hand, Satoyama has been used, but if humans will not interfere Satoyama, it will be desolate. Due to the energy devolution in recent years, Satoyama has become no longer beneficial to humans. So there is no inevitability to be involved, and the number of neglect has increased. Satoyama has two such aspects. Next, about Satoyama's problem in recent years, the biggest problem is people's indifference to Satoyama. Lack of the need for people to get involved with Satoyama causes a decrease in people's interest in Satoyama. It leads to the neglect of Satoyama. In the neglected Satoyama, the transition progresses and the number of evergreen trees increases, and the closer approach to the polar forest leads to the decrease in biodiversity. There are also other problems of depopulation, aging, and urbanization. What we humans can do to protect them against these problems is to cut down trees to stop the transition of Satoyama. In addition, in order to prevent the decline of Satoyama due to people's indifference, it is necessary to educate people to pay attention to them. Next is about the actual activity.
Next, we would like to talk about the activities we actually participated in. We took part in on-site maintenance work in Satoyama. So this experience, we learned that simply knowing
something is not enough. Doing it ourselves was completely different. For example, these are correct methods for cutting down trees. If done incorrectly, it can cause serious harm to the ecosystem. We also realized how important the knowledge and experience of local people are. Their techniques are not just traditions, they are sustainable practices rooted in experience. The activity helped us understand both the weight of responsibility and the involved in environmental conservation. At the same time, we also discovered many challenges related on Satoyama conservation. To be honest, we felt that continuing this kind of work without any compensation would be difficult. During this kind of labor every weekend is not easy. There are serious issues such as a shortage of workers, lack of technical support, and over reliance on unpaid volunteer work. We come to realize that goodwill alone is not enough to sustain Satayama. Without proper support and a sustainable system, this variable ecosystem may be lost. This experience made us reflect on how we as a society can support these who protect nature. From the outside, these activities may look beautiful, but in reality, they involve a lot of hard work and unspoken challenges. We truly felt this gap between appearance and reality. If nothing changes, it will become harder to continue maintenance and management. The natural environment itself is at risk. Behind what appears to be beautiful work, these are deep structural issues. That is why we believe a sustainable system must be built. One that is supported by the efforts of many people.
At the same, I will talk about Satayama's conservation and ecosystem services. In the past, people used natural resources without considering the environmental impact. For example, wood was a necessary material in daily life, like for building houses or making fires. Now, necessary of Satayama has disappeared. We must define the meaning of our involvement, such as why we are involved and what the purpose of conservation is. Therefore, it is important to make services in which people gain something at the same time people protect nature. In other words, the relationship between nature and people is a win-win relationship. That's where ecosystem services come in. Ecosystem services are the benefits that nature provides to people. There are four main types of ecosystem services. First, supply service. It means nature provides us with food, water, fuel, and medicine. Second, adjustment service. It helps control nature events such as limiting disasters and diseases, and regulating climate and water. Third, culture service. Nature gives us beauty and mental satisfaction. Many people enjoy hiking, sightseeing, or relaxing in nature and natural places. Fourth, basic services. It is the basic service that support all on Earth, such as soil formation, nutrient cycling, and oxygen production through photosynthesis. These services are deeply connected to our daily lives. In Japan, for example, farmers grow important crops that depend on healthy ecosystems. Without pollinators like bees or clean water, crops would not grow well. To continue receiving these benefits, we need to conserve biodiversity and protect ecosystems. We insist a win-win relationship is the important keyword to protect Satayama. We must pay attention to how we interact with Satayama and take action to protect it.
To sustain Satoyama landscapes in the modern era, three key elements must work together. First is local residents. They possess deep-rooted knowledge of the land and traditions. Their continuous involvement in land management and cultural practices forms the foundation of Satoyama. Second is external participants. These include people from outside the community, such as students, urban residents, and volunteers, who bring fresh energy and ideas. They help bridge the gap between local nature and modern society. Third is institutional and financial support. Policies, systems, and funding mechanisms are essential to maintain long-term activities. Without stable support, effort by individuals and communities may not be sustained. These three elements must not exist in isolation. They need to interact and reinforce each other. Only then can we build a truly sustainable human and nature relationship. Here, we will explain the interrelationship between each of these elements. First, we will explain the relationship between local residents and external participants. External expertise enhances the capabilities of local residents, while the traditional knowledge and on-site participation of local residents strengthen external initiatives. Next, we will explain the relationship between local residents and institutional and financial support. Institutions define local residents' rights to land and resources, and financial support provides incentive for Satoyama conservation and strengthens ownership rights. Finally, we will explain the relationship between external participants and institutional and financial support. External participants act as intermediaries between local needs and government policies. Sansinri is a rural village located within one national park in Korea. Once facing depopulation and environmental degradation, the community revitalized itself through innovative effort. Supported by government funding, they cultivated wild plants on abundant farmland, promoted cultural festivals like pottery and local theater, and invited visitors through rural areas, a rural experience program. While newcomers from nearby cities now live here, the interaction between old and new residents remains limited. Still, Sansenri shows how traditional practices, tourism, and policy support can breathe new life into Satoyama. Another example is the sustainable use of forest by indigenous people and the indigenous forestry system in Canada. Indigenous people make a living by managing timber, hunting, and fishing, and within the limit of their national resilience. Canada's Indigenous people use the resources to meet their cultural and spiritual needs. Another system of partnership between First Nations and the federal government is the Aboriginal Forestry Program. This system was created to improve the economic conditions of the indigenous people. It has enabled indigenous peoples to create new business by combining traditional skills with new concepts and technologies.
So what is essential for Satoyama conservation today? Our answer is clear: rebuilding a mutually beneficial relationship between people and nature. Without people, Satoyama cannot survive, but without value for people, they will not stay involved. Win-win is the only path forward. This win-win relationship depends on three essential elements: local residents with knowledge, roots, and cultural connection; external participants, students, volunteers, urban citizens; institutional and financial support to ensure long-term sustainability. These three must work together in continuous balance. We found inspiring real-world examples: Sanshin Lee Korea, branding, traditional tourism, and ecology. Canada is indigenous forest stewardship supported by law. These cases show how the three elements can work together. Satoyama is no longer just a local issue. It stands at the forefront of global environmental thinking. From Japan to the world, it offers model of coexistence, a reminder that sustainability begins with relationship. Thank you for listening. Let's rethink how we live with nature. That concludes my presentation. Now I'd like to open it up for questions.
Okay, thank you very much. Yeah, so at moment. So I think I'm going to open for discussion, and please you look here. Yeah. Okay. Okay. So I have some message from the UMMUL concerning just for clarification. Yeah, regarding the what is it, the cat dying. Yeah. So they said that the allow us to respond regarding the duration of natural dyes, which is currently a problem because they are not long lasting. However, we are trying to address these issues through research and natural dyes. So it's mean the synthetic still a little bit what is it, durable is longer than the that one, but we are going to they are going to what is it, to make us can compete with the synthetic one. Okay, thank you very much for your confirmation. Yeah. So also now I am going to give because actually time is over, but it's kind of the bonus yeah, from our from the committee. So I'm going to maybe just two questions. Maybe I will give to the audience. Okay. Please directly or raise your hand and you can ask concerning what is the essential for sustainable Satoyama conservation? So hopefully there's some question. Maybe some one of the student before you coming to Japan, maybe you can ask in the advance. Can you hear my? Anyone would like to ask? Okay, so maybe I will start to ask maybe. Okay. why in this case some people neglect Satoyama. So I asked this one. In my case, actually, I don't want to neglect this one. But why it's happened in Japan? They neglect the Satoyama. You can directly to ask to answer this one.
Because the Satoyama should provide the fuel and food for people, so and people treats the mountain Satoyama. So that's the old relationship, but it's broken, the relationship, because there is oil and there is electricity, so we don't need to use the Satoyama. So that relationship is broken. So we ignore the Satoyama.
Okay, thank you very much. So it's mean some kind of the competition, yeah, between the modern lifestyle and then the Satoyama. So actually, I would like to ask again, but anyone would like to ask maybe because we still have one question? Student from Kalimantan, from UMMUL, or maybe from Diponegoro, or maybe from Sudirman University, Udayana University, and also from UiTM and Pajajaran Bandung. Okay, so I think I will ask again. Yeah. So therefore, how to stimulate the young generation? In this case, they would like to involve with this kind of because I think we must maintain the Saitoyama in this case. So any kind of the idea, maybe you can repeat again. Maybe you just know, maybe you already explained this one, but which one the important thing in this case to invite the young generation? Don't talk on only this decade, yeah, but also maybe for the future, yeah. Thank you. So please answer. How to stimulate and how to what the problem solve. So yeah, young generation can also would like to maintain, yeah, and an idea.
And that is discourse, because we involve and or discourse involves Satoyama.
Yeah.
And so we didn't know that Satoyama before receiving this cross, but I received this cross, so I know this Satoyamas are currently things. So this cross should more spread or every or area, so every university.
Yeah.
So we should continue to hold this cross for future.
Okay. Thank you very much. So it's mean what this KGU, they already initiate this one, so you can continue, and also we hope one day we involve also local people. I mean the local people and the other people from the other place to invite and then make kind of the for example ecotourism and the other yeah. So it's mean like that. So I think okay, thank you very much. So I think I would like to say thank you very much for KGU Group 3. So I think we are going to close. Yeah. Thank you very much. So would you like to close your share? Yeah. Would you like to close your share? Okay. I think I would like to give some final talk. Yeah. because I just know as in the beginning, I already informed, please open your camera, yeah, and also some kind of the information just now from To-sensei because he has some problem with his body, so he returned back early. So now I'm going to give some final talk. So I would like to say thank you very much, yeah, to all participants, yeah, and also to all deans from several university, yeah, several faculty also, yeah, and also I would like to say thank you very much to contact person, yeah, and also some supervisor in this case, and particularly, I would like to say thank you very much for all participants. Yeah, already have some effort in this case because I heard that in your university also this is the holiday. Yeah, and but you still, I would like to say that help and you continue and join with this program. So, actually, I really enjoy all this presentation. Yeah, based on the, I mean, on behalf To-sensei as the chairman, and now I'm as the assistant. And also, I'm very glad to say that if you are also enjoy this kind of the event, and because you already share a lot of information from your university that we cannot reach from Japan, come to the Kalimantan, and from Kalimantan would like to know about the other area like in Java or and also in Malaysia and vice versa from Malaysia to Japan and also to Indonesia. So, but hopefully, in the future we have such kind of the chance, yeah, we can visit. So therefore, we hope you also enjoy with this some presentation, this kind of the workshop, and as you know that this is the fifth workshop, yeah, and you know that we already start this workshop, I think starting when we have such kind of the pandemic, yeah, in the era pandemic. So we create this Zoom, and then now in the fifth. So hopefully next we will have the sixth of the workshop, and so we are going also to expand because for this year, I think the first time for from the faculty biology faculty, what is it, Mathematica and Natural Sciences from Udayana the first time, yeah. But the other faculty already maybe the second or the third, yeah, and the fourth. So therefore, thank you very much for your joining from the what is it biology? I mean Department of Biology, but the Faculty of Mathematics and Natural Science Udayana University. So therefore, hopefully in the future we are going to also to extend the number of the university that we can more share and we can what is it, we can have some new information, yeah, from the other area, yeah, that we cannot reach that area. And then also I hope we can meet again, yeah. I don't know, maybe one day I can visit Malaysia or you visit to to Japan, and also we visit to Indonesia, yeah. Honestly, this month I'm going to visit to Bali, to Dayana, in order to make some kind of the survey. So, but yeah, hopefully we will meet again. And maybe I would like to say that today, thank you very much. And maybe I am going to close. Yeah. See you later. But before that, as I inform you, please open your camera. I'm going to make pictures. Okay. Okay. I think first, yeah, 1, 2, 3. Yeah. 1, 2, 3. Okay. And 1, 2, 3. Okay. Thank you very much. So I will close, and thank you very much to everybody here. To what is it? We call this Mr. Secha or Professor Secha. Yeah. And also maybe Retno.
Thank you.
Thank you. Yeah. Thank you very much for Professor Eni Rosama, yeah, also.
Professor Eni Rosama.
Yeah, Eni Rosama, yeah.
Long time not to see you.
Hello, yes.
And thank you for all participants. So we close today, yeah. And see you later, maybe next year. Okay, thank you very much.
Mata
Halo, Mas
Rainen.
Despren.
Mata Rainen,
Profesor
okay, Mata
Seja,
Rainen.
ya,
Thank
Dr.
you,
Dedin.
Eni,
Yeah,
for
Mata Teten, yeah.
Thank you very much,
Thank you
yes,
very
for
much,
inviting
yeah.
us.
Thank you, everybody.
Okay.
Thank you.
Halo, halo, halo.
Okay.
Halo, see you in Samarinda ya.
Yeah,
Yeah,
see
see you
you.
in Samarinda.
Profesor
Berapa nih?
Seja, ya. Oke.
Thank you. Assalamualaikum.
Assalamualaikum.
Waalaikumsalam.